So this theme will be presented in 4 main parts, namely the peer project, objectives, work plan, and purpose, besides the composting experiment, then the assessment of the produced compost quality on organic barley, and finally, the expression of nutrient transporter genes related to the effect of the use of compost. So climate change could damage the agriculture, the agriculture sector, or reduction in crop yield, over cost on food marketing, increased extent of pest invasion, restrained water availability, aggravation of drought periods, reduction in soil fertility, low livestock productivity, and high production costs. These considerations are valid at the global, continental, and local scales, with very crucial conditions in Tunisia known by deficient organic stock in soil, about only 0.5% that are very poor in nitrogen, phosphorus, potassium element, and plus a massive and uncontrolled use of chemicals. So our current concern is how can we have an acceptable agriculture production. To come over this situation, we have to analyze what we have as useful resources and what can be done with them. The analysis shows that in one hand, we have very degraded soils, and on the other side, we have a huge amounts of waste. So the solution that we suggest is to proceed with the composting and to disseminate this useful process for farmers. In this context fits the peer project presented in more details in the next slide. So the project is titled Developing Organic Soil Management Technologies to Enhance Carbon Capture, Climate Adaptability, and Sustainability of Smallholder Farms in Tunisia. And the main objectives are integrated soil management into crop rotations, adapted local conditions to improve climate adaptability and sustainability in Tunisia through an appropriate composting methods and recovery of residues and organic waste from small farmers. The realization of the project was celebrated in 2 phases. The first one called explanatory phase. So we proceed with a survey with smallholder farmers in the south of Tunisia. Then we did the compost windrow. And finally, we studied the effect of compost on the production parameters. And those results fit and complete the second phase, named identification of compost microbial community. The following part will be dedicated to explaining the experimentation of composting in Cheni location in the south of Tunisia and the results that come out of it. Starting with the windrow composition and design. Thus, the composition of windrow number 1, growing from bottom to top, is wood chips 15%, sheep manure 35%, wood chips 15%, and sheep manure 35%. The second one contains wood chips 15%, soaked in water, and cow dung, sheep manure 35%, and wood chips 15% soaked in water and cow dung, and sheep manure 35%. And the third one contains a combination with wood chips at 30% and 70% of sheep manure. The physico-chemical parameters that have been evaluated are pH, electrical conductivity, humidity, total organic carbon, organic matter, temperature, and dry density. The microbiological parameters are total aerobic mesophyll count. We measure bacteria and fungi, fecal pollution indicators, bacteria, and screening for the presence of pathogens, bacteria like Salmonella or Pseudomonas. From a microbiological analysis, the final products produced in the 3 studied areas have a load of fecal pollution indicators in accordance with the national and international standard. With the produced compost can be so the produced compost can be used in agriculture without risk. We didn't find pathogenic bacteria in the final products for all studied area in accordance with the national and international standard. So the produced compost can be used in agriculture without risk. Following is the assessment of the produced compost quality on organic barley, subdivided in 2 parts: seed germination and production and nutritional quality. Here is a protocol of the different treatments contains compost with compost tea applied on the barley cultivation. Here, some illustration on photos, starting with the collection of waste from the oasis, their crushing, soaking in water and cow dung, until the installation of the compost windrows. And here we see other photos that illustrate the use of compost tea on barley cultivation. For the seed germination, we used an aerated compost extract preparation at 1:10 ratio, and with a determination of the effect of compost extracted on barley seed germination with 25, 50, 75, and 100%. And measured parameters are germination percentage, relative seed germination, relative root growth, germination index, and seedling vigor index. For the growth assay in the glass house, plastic pots with capacity of 5 liters were used with 5 levels of compost from 10 to 50 tons per hectare, and the control without compost. Now, assessed parameters are agronomic and physiologic parameters and mineral analysis. So here the doses of 25% and the 75% extract gave a germination rate of 94%. Here, the highest value of the germination index and the vigor index were obtained by the dose of 25, 50, and 75%. And the relative seed germination is equal to 100% for 25% dose. And the greatest root elongation was recorded in the seedling treated with the 25% and 50% dose. Here, the compost didn't negatively affect the germination percentage for the different concentration. In general, the highest photosynthetic activity, stomatal conductances, and intercellular carbon dioxide concentration and transpiration were recorded in the treatment number 2 with 20 tons per hectare, and the treatment number 3 with 30 tons per hectare. Here, the highest height value was obtained using 30 tons per hectare dose. The maximum for root elongation is observed with the 10 ton per hectare dose. And the shoot and root biomass increased simultaneously with the increase in the doses provided. The use of compost improved the total chlorophyll contains compared to the control without compost. And the use of high doses of 40 tons per hectare and 50 tons per hectare reduced the concentration of carotenoids and chlorophyll B. Compared to the control, crude protein decreased under the effect of the different concentrations of compost. Following is the assessment of the effect of compost on expression of nutrient transporter genes. So the experiment design in the field was done in 4 treatments. The treatment number 1 is the control without compost. The T2 is the compost extract. The treatment number 3 is with compost with 30 tons per hectare. And the treatment number 4 is the combination of compost and its extract. The first step was to determine in silico the list of primers specific for the 25 genes coding for the transporters of nitrate, phosphate, and ammonium. Then we moved to total RNA extraction. The majority of the genes studied were differentially expressed according to the organ and according to the different treatments applied. And the induction of the expression of several genes following the addition of compost reflects an improvement in the absorption of essential minerals such as ammonium and phosphorus contents in barley leaves and roots. So to sum up, we return to the following facts. All the compost produced during our study are rich in organic matter. They have level content ranged between 45% and 54% with high level of nitrogen, phosphorus, and potassium, and they are microbiologically healthy. Overall, our produced composts meet the national and the international standards, and in all studied area, produced compost can be used in agriculture without risk. Now, this practice will have several beneficial effects on soil and on the plants. The phytotoxicity analysis of the compost and its extract have shown the compost was free of any phytotoxic effect, mature and stable, and can be used with complete safety according to the results. Those corresponding to 30 tons per hectare is the appropriate for achieve the greatest improvement in barley plant growth. And transcriptomic analysis highlights the ability of compost to provide the nutrients needed for barley growth. As main recommendations, we conclude that co-composting date palm waste could be an effective and environmentally friendly approach in terms of date palm waste management. And this approach improves biological fertilization of South Tunisia organic matter deficient soils by their content in micro and macronutrients. And this compost could be a good biofertilizer for other crops in the 3rd stage, especially fodder crops, and could replace chemical fertilization with satisfactory results in the oasis of Chen. And thank you for your attention.